Today we're going to look at the various divisions of the nervous system, and then we're also going to do an overview of a nerve impulse and how it propagates within a neuron. So as animals become more complex, so do their nervous systems. So whether we're comparing the nerve net of the cnidarian to the associative neurons, which are kind of a primitive brain in the flatworm, which is an example of a rudimentary central nervous system, and all of the subsequent evolutionary changes in nervous systems, we can see the development of elaborate brains and sensory systems involved in the coordination of various functions of the organism. And we can think of the central nervous system as the command center of the vertebrate nervous system. And it's composed of the brain and the spinal cord. It receives stimulus from the sensory portion of the nervous system via sensory neurons and sends impulses that influence muscles and glands via motor or effector neurons. These impulses from the CNS will stimulate effector organs, whether voluntary skeletal muscles of the somatic nervous system or involuntary responses in the autonomic nervous system, such as involuntary muscle tissue, including the heart or smooth muscle tissue of the walls of our arteries, or glands. And in the autonomic nervous system, we see a further division between the sympathetic nervous system involved in fight or flight response and the parasympathetic nervous system, which controls the body's organs in a resting state. And we can look at the primitive fish brain to examine the basic organization of the vertebrate brain. And the three basic divisions of the vertebrate brain are the hindbrain, the midbrain, and the forebrain. And in these divisions, we see in the forebrain the cerebrum, the thalamus, and the hypothalamus, the midbrain, which in this primitive model, the structure indicated is the optic tectum, which is involved in visual processing. And the hindbrain, we see the cerebellum, the pons, which is not actually indicated on this diagram, and also the medulla oblongata. Now, in contrast, in the brain of the mammal, we see a much more developed cerebrum, but the same basic level of organization can be seen with the hindbrain, midbrain, and forebrain. And as a reminder, I would like you to refer to the video that we posted for the Nervous System Lab to reference brain structure and function of mammals. And along with the brain, the other component of the central nervous system is the spinal cord. And the spinal cord is basically a cable of neurons that extend from the brain down through the backbone. It's encased in the vertebral column, and it's also covered in membranes called meninges, and there's more detail about the meninges in the lab video. And messages from the body and the brain run up and down the spinal cord. It can be thought of as the information highway of the body. But in addition to relaying those messages, the spinal cord is also involved in reflexes, which are the sudden involuntary movement of muscles. And before we take a closer look at how neurons actually function, this slide is just to remind us that we do have sensory neurons that send impulses from sensory receptors to the central nervous system. And we have motor neurons that conduct impulses from the CNS to the muscles or to glands, basically to effector organs. And we have interneurons, which are neurons that are located in the central nervous system. Now, the stimulus and response that's the simplest to review is the monosynaptic reflex. The monosynaptic reflex involves a sensory neuron and a motor neuron, and there is just one synapse, hence the name monosynaptic, that the impulse needs to cross between its initiation point at the sensory receptors and its destination at the effector organ. However, we should keep in mind that most reflex responses also involve an interneuron, such as seen on the right, where the interneuron is the intermediate between the sensory or afferent neuron and the motor or efferent neuron. And the basic structure of a typical neuron consists of dendrites, which are the branch-like projections that extend from the cell body. These are going to be the structures that carry a stimulus from the external environment into the neuron. And the dendrites are connected to the cell body, which is where the nucleus of the cell is. And then the long extension that we see coming out, at least in this picture, at the base of the cell body is the axon. And this axon in particular is myelinated, which means it's covered in a myelin sheath. The myelin sheath is actually made up of Schwann cells that are wrapped around the axon. And we see that there's a gap in between Schwann cells along the axon that's labeled the node of Ranvier, which is where action potentials take place in a myelinated axon. The mechanism involved in the propagation of a nerve stimulus is built around the fact that there is an electrical difference that exists across the plasma membrane of these cells. So any separation of electrical charges of opposite sign represents an electric potential that is capable of doing work. And this difference in charge is based on the concentration of positively charged ions and negatively charged ions that are outside and inside the cell. And the outside of the cell is going to be more positively charged compared to the inside of the cell. And if we were to look at the difference between the outside portion and the inside portion, a typical difference would be around negative 70 millivolts. And this can actually be considered kind of like a battery, whereas you would have a positive pole would be represented by the external surface of the plasma membrane, and the negative pole would be represented by the interior leaflet or the interior portion of the plasma membrane. And it's the permeability of the plasma membrane to the various positively and negatively charged ions that creates and maintains this electrical gradient or this electrical potential. And the primary membrane structures responsible for maintaining this gradient are the sodium-potassium pump and also sodium and potassium leak channels. Now, the sodium-potassium pump shuttles sodium ions from the interior of the cell to the exterior of the cell, and it shuttles potassium ions from the exterior to the interior. The ratio in which it does that is for every 2 potassium ions that it moves into the cell, it transfers 3 sodium ions out of the cell. So this, of course, results in a difference in charge from the exterior to the interior. Additionally, the potassium and sodium leak channels are embedded in the cell membrane at different concentrations. There are actually more potassium leak channels in the membrane, which basically means that the membrane is more permeable to potassium, which means that more potassium will leak out of the cell through its leak channels than sodium will leak into the cell through its leak channels. And we're going to use this slide showing how ligand-gated channels function as an example of how a nerve impulse initiates in the neuron. So there's going to be some sort of external stimulus, where in this case what we're seeing is a synaptic cleft where we see a presynaptic neuron that's releasing a neurotransmitter. In this specific example, it's acetylcholine. And what the acetylcholine is doing is it's binding to receptors on the postsynaptic cell membrane. So we can think of the postsynaptic cell membrane as being the membrane of our neuron at the dendrite. And when this neurotransmitter binds the receptors that are embedded in the cell membrane of our dendrites, it triggers the opening of these channels that allow sodium to enter into the cell. And sodium, being positively charged, is going to actually change the membrane potential or the charge difference between the outside and inside of the cell at this region. And this influx of positively charged ions into the more negatively charged interior of the cell is going to cause depolarization, basically meaning that the charge difference between the interior of the cell and the exterior of the cell is going to decrease. And we call this influx of sodium ions at the dendrite in response to external stimulus a graded potential. And so this potential is going to vary based on the number of receptors in the dendrites that are being opened, which is going to be related to the strength of the external stimulus. Now, these sodium ions that enter the neuron travel down the dendrites towards the cell body. And if enough sodium enters the cell body and travels across to a region called the axon hillock, we will end up with the creation of an action potential, which an action potential is going to be our actual nerve impulse that's going to propagate down the entire axon of the neuron to the axon terminal. And a typical threshold for whether or not an action potential will take place is negative 55 millivolts. So in other words, if enough sodium ions reach the axon hillock to change the membrane potential at the axon hillock from negative 70 millivolts to negative 55 millivolts, that will result in the creation of an action potential, or in other words, the creation and propagation of a nerve impulse. Now, once the graded potentials result in a depolarization to threshold at negative 55 millivolts at the axon hillock, voltage-gated sodium channels in the local area of the hillock open. This causes a rapid influx of sodium ions into this portion of the cell, and this causes additional localized depolarization, or as the book calls, the rising phase. Once a maximal voltage is reached, inactivation gates on the sodium channels close, blocking further influx of sodium. At the same time, voltage-gated potassium channels open and potassium moves out of the cell, which results in a repolarizing of the membrane potential, which the book refers to as the falling phase. During this falling phase, depolarization cannot be triggered in this portion of the axon since the sodium channels are still inactivated. After repolarization is sufficient, the gates of the potassium channels close and resting potential to negative 70 millivolts is restored by the sodium-potassium pump and the sodium and potassium leak channels. Now, this action potential propagates from the axon hillock to the axon terminal. But the way that it does that is that it actually takes place over and over and over again in adjacent portions of the axon. As you remember, the initial influx of sodium ions at the beginning of the first action potential, those sodium ions diffuse into adjacent portions of the cell or adjacent portions of the axon, which then triggers an action potential in that region, which causes an influx of sodium, which diffuses to the next portion of the axon, which triggers another action potential there as well. Now, the reason why an action potential propagates unidirectionally is due to the fact that during the repolarization phase in one specific portion of the axon, those sodium channels are inactivated, so we cannot have an additional action potential take place in the region previous to the one where it is currently taking place. So as you can see by this image, what takes place is an action potential happens to the leftward region of the axon, according to this picture, and then that triggers one in the next portion of the axon to the right, which then triggers one in the next portion of the axon to the right of that. And then once that action potential reaches the axon terminal, that triggers an influx of calcium ions at the terminal, which triggers the fusion of vesicles that are full of neurotransmitters to the cell membrane at the axon terminal. These neurotransmitters are then released from the cell via exocytosis. And depending on where this is taking place, either those neurotransmitters are going to bind receptors in effector organs, or they will bind receptors in dendrites of the postsynaptic neuron, thereby triggering the intake of sodium in that neuron and repeating this entire process all over again in another neuron. Now, the way that I described the propagation of an action potential earlier in this video was specific to unmyelinated axons. And a myelinated axon actually propagates a nerve impulse more quickly than an unmyelinated axon. And this is due to the fact that the action potential only needs to take place at the nodes of Ranvier. And in between the nodes of Ranvier, within the myelinated region of the axon, the sodium is able to quickly diffuse across the myelinated portion, and this is due to a decrease in capacitance resulting from the insulative quality of the myelin sheath. And this effect is called saltatory conduction. So today we took a look at the nervous system in general, looking at the central nervous system and components of the peripheral nervous system, including sensory neurons and motor neurons, or afferent and efferent neurons, and also the subdivisions of the somatic and autonomic nervous systems. Additionally to that, we took a look at the nerve impulse, looked at some differences between graded and action potentials, some of the events involved in the creation of an action potential, some of the steps involved in the action potential, and also the ways in which the action potential is propagated through repeated events along the length of the axon of the nerve cell.